Morning students. Today we are going to see about translation in this video. So the translation process is also known as protein synthesis. So here the genetic material which is stored in the DNA was first initially transcribed into mRNA copies, and from that mRNA copies the language of mRNA is translated into proteins, that is amino acids. So that is the process we are seeing under translation. So this is the assembly how the translation process takes place. So the process of converting the nucleotide code, that is the nitrogenous bases present in the mRNA, so which is the transcript or a copy formed from the DNA. So this will be converted into amino acids. So this process of translating this language into a language of amino acids is known as translation, and the direction in which the translation takes place is 5' to 3' direction, and this mRNA is read or interfaced by tRNA, transfer RNA, and this transfer RNA has an amino acid attached with that. So based on the code present here, it assembles the amino acids to form the protein needed. So that is what happening in the ribosome, and this is the structure of ribosome. It has a larger complex and smaller complex. So a ribosome with the transfer RNA and mRNA is present in this assembly of protein synthesis or the translation process. So what are the components needed for a protein synthesis or translation process? First, the mRNA, that is the blueprint. So from where we are getting this mRNA? By means of transcription from DNA. So DNA undergoes transcription process, then it will be modified, post-transcriptional modification is done, and the final blueprint, the mRNA, was produced, and it will be transferred from the nucleus through the nuclear pores into the cytoplasm. So in the cytoplasm we have the ribosome. This is the factory where the protein synthesis is taking place, and to read the mRNA or to decode the mRNA we need an interface, that is tRNA. So it acts as an adapter. It has a three anticodon zones. So this is the anticodon region. So it attaches with the codes present in the mRNA, and here the amino acid will be attached. So based on the code, the amino acid will be assembled in the factory. So this is the main factory unit for protein synthesis. So you can imagine this like this. So this DNA is like if you take you have a soft copy of a medical textbook. So it has thousands of pages. So you want only one chapter to be printed out as a hard copy. So what you are doing, you are extracting that pages alone as a separate PDF. So that is known as transcription. So you are taking a small copy from this a large file. So that is transcription. So you are taking that file in a pen drive and you are going to a printing center. So there you are giving that pen drive, and that pen drive will be inserted into their computer and it will be read by their system. So that is known as tRNA. So this tRNA extracts the information from this pen drive or from this blueprint copy, and it transfers to the printer. So from here the information present in the printer is transferred, present in the uh, pen drive is transferred through this interface. So this is the tRNA, and it will be transferred to the ribosome, that is the printer. So the printer has ink. It has some forms of certain inks. Based on the data present here, it will assemble the inks and it will produce the final product, that is the printed copy of that particular chapter. So like you can imagine this example to understand the process of translation. Likewise, you are getting a small copy from this, the copy which is needed for the protein synthesis from the whole genome as a transcript, and it will be transferred to the cytoplasm, and in the ribosome it will be read in ribosome by the interface tRNA, and the tRNA has, like the ink in the printer, it has a fixed amino acids here. So based on the code present in the mRNA, it will assemble the amino acids and thereby it will produce the protein. So certain protocols which are followed while reading the genetic code. So usually for an amino acid to be formed, it needs 3 nitrogenous bases, that is a triplet codon. It is known as a triplet codon. So for a single amino acid, for example, if you take this proline, it can have these kind of combinations. So it can have cytidine, uracil. At the end, it can have these different types of amino acids. So this triplet also forms proline. CUC also forms proline. So you can see the first 2 letters are almost fixed and the last letter is changing, but this change is not affecting. So even though it is changed, some form of change is allowed here, and this triplet codes for a single amino acid. Likewise, you can see for all other amino acids, you can see GGU codes for glycine and GGG codes for glycine. Likewise, so this form of assembled or this form of genetic code is followed. These kind of protocols is followed. So we have to remember certain terms regarding the protocols of reading genetic code. First thing is universality. So if you see a codon, for example GAG, so if it codes for a particular amino acid in one organism, it is same for nearly all living organisms from bacteria to humans, that is universality. And next is degeneracy. What is degeneracy? Multiple codons can do or can code for exact same amino acids as we have seen here. So here we can see for proline we can have 3 types of codons, but the difference is only at the last amino acid, that is the third nitrogenous base. So this property is known as degeneracy or redundancy. So you have to remember this to understand what is wobble hypothesis, that you will see in the following slides. And then non-overlapping. So when you are, for example, if you see this is the mRNA codons, you are reading from 5' to 3'N. So if you read these 3 nitrogenous bases, that is one codon for a particular amino acid, and you will not take, or the body's translation process will not overlap, or it will not read the next 3 as like this. It will read, it will skip to the next codon and it will skip to the next codon. So there will not be any overlapping while reading. It will be a distinct codon. So the triplet codons are read continuously without skipping or sharing bases between adjacent groups. So there will not be sharing between these 2 codons. So if this codon is read, these 3 nitrogenous bases will be read, and for this codon, these 3 it is a distinct one. So a distinct amino acid will be formed. Okay. So these 3 are the common protocols or common basis for reading a genetic code. So first is universality. All organisms give a specific amino acid for a particular codon. That it will not be vary depending on the organisms. The only exception is mitochondrial DNA. When it is translated, certain codes will be changed. So other than that, all codes will be universal. So degeneracy, multiple codons can code for exact same amino acid with flexibility at the last nitrogenous base. Okay. And non-overlapping, codons will be read distinctly. There will not be any sharing between adjacent codons. Okay. So this we have seen about redundancy or degeneracy in the previous slide. That is, the last nitrogenous base can be changed so that results in a forming of multiple codons coding for a single amino acid. Okay. So how it is happening? It is explained by a wobble phenomenon. So in the first two codons, that is, a codon is made up of three nitrogenous bases, 1, 2 and 3, and first two nitrogenous bases require strict, so they bind strictly, the base pairing between, so this is the mRNA, this is the nitrogenous bases present in the mRNA, and this is the anticodon arm of tRNA. So when the base pairing is taking place between the codon present in the mRNA and the anticodon present in the tRNA, the base pairing is very strict. The spatial tolerance or the binding interface is in a strict log position in the first two bases. So there will not be much change in the first two positions. Whereas the third base, it permits a spatial tolerance. There will be a flexible spatial geometry. So it acts as a molecular hinge. It can rotate and it can have a varied base pairing. So either C can pair here or it can pair here. So you can see here. So a various form of spatial geometry help in the redundancy in the last, that is third nitrogenous base. So this leads to the redundancy or degeneracy property of genetic code. So that is for the codon. So that leads to multiple codon coding for a single amino acid. So this is explained by the wobble phenomenon. So let's see some things about tRNA. So the tRNA basically it coordinates the code and it brings the corresponding amino acid. So how it is bringing? So it has an anticodon loop. So this anticodon loop has a complementary sequence to the mRNA code. That's why we are calling it as anticodon loop. Okay. And this is known as acceptor stem. It is a 3' so here it accepts the amino acid. So it also has a DHUAM, so that detects the particular specified amino acyl transferase. That enzyme helps in the binding of amino acid at the 3' end. So for example, if this tRNA needs a phenylalanine amino acid, that DHUAM, it detects the phenylalanine amino acyl transferase. So that helps in the binding of that amino acid at the 3' end. So when a codon for this phenylalanine is read by the anticodon arm in the mRNA, this phenylalanine will be incorporated or assembled in the protein synthesis that we will see in the following slides. So what is the first step in translation? So the first step is charging of tRNA. So the tRNA is present in the naive form or in a normal form. It should be charged with an amino acid. So an amino acid is attached to the tRNA by the enzyme amino acyl tRNA synthetase. So that is known as amino acyl tRNA. So this reaction is known as charging of tRNA, and that is the preparatory first step for translation. So for this we need energy. It is a highly energy consuming process. It utilizes ATP, and this energy usage is a strategic point. How this will be utilized in the future peptide bond formation for protein synthesis, that will be seeing later. So this step is known as charging of tRNA. Since this energy is conserved, so we are prepaying the energy. So we are doing a prepaid recharge here. So this energy will be utilized later. So that is a strategy for using this energy here. Okay. So it is a preparatory step, the charging of tRNA. Let's see the structure of ribosome, the architecture of ribosome to understand the future steps. So a ribosome has a larger subunit and a smaller subunit, and it has three sites. One is A site, that is acceptor workstation, P site, that is peptidyl workstation, and E site, that is exit workstation. What is these three stations? The acceptor station, it binds the incoming tRNA. So the tRNA, it has the corresponding amino acids. So here the mRNA is present here while translation. So based on the code, it will accept the tRNA with the particular amino acid, and it will be transferred to the peptide site where it will hold the polypeptide chain. So that will hold while the protein synthesis is taking place. So for that, this is known as the peptidyl workstation. So it anchors the tRNA holding the growing polypeptide chain. And what happens in the exit site? Once the acceptor site, it accepts the new amino acid, it will be holded by this peptidyl workstation, and the formed amino acid will be transferred to this newly forming protein chain. So the peptide bond formation is taking place, and this tRNA will become free, and it will move to the exit site, and from there it will be sent out. So it will be sent for recycling. So here in the exit site, the deacetylated tRNA, that is the empty tRNA, will be ejected from this factory. So this is the factory setup. Here accepting is taking place, here holding is taking place, here the exit process is taking place. So the ribosomal is a workstation for the protein synthesis. So in the first step, we have seen the charging of tRNA. Next, what is happening in the second step? So it is initiation. So first, the complex initiation at the start codon. So what happens? First, the mRNA, it should be scanned for an initiating region, that is a code like AUG. So it is a start codon. So this codon should be detected. For that, a scanning process will take place. So who is doing the scanning process? It will be done by this smaller subunit of ribosome. So it will be doing the scanning process, and it will find the AUG. So for this, an initiation complex need to be formed. So how this initiation complex is forming? By the help of initiation factors. So EIF means eukaryotic initiation factor. So this initiation factor binds with the cap region. As we see already, after transcription, in the post transcriptional modification, we'll have a cap formation, that is 7-methyl guanosine cap formation. So that will act as an ID card. So this ID card will be recognized by this initiation factors, and it will be binding with the cap and forming the cap binding complex. And after this, scanning is taking place, and the AUG region, that is a start codon, is recognized. And the speciality about this start codon is it will not go for the acceptor site initially. It will go directly to the peptidyl site, and it will be locked. So it will be attached here, and when it is attached here, this larger complex also attached to the ribosome, and this entire process is known as initiation complex. So an initiation complex will be formed. So this is the steps taking place in the initiation. So as we see here, the corresponding tRNA with the anticodon arm is attached here, and it has the amino acid methionine. So what is the next step? We have charged the tRNA, it was bound, and it was initiated as a initiation complex. So now it is elongation. So the amino acid need to be added further. So the initiation complex, that is a start codon, directly attaches to the P station, that is a peptidyl station, and it has an amino acid attached to it. So now the acceptor site is free. So what it does, it accepts the next amino acid with the corresponding codon present here. So here one codon is there. The next codon is available in the mRNA near the acceptor site. So based on this codon, it will accept the next amino acyl tRNA. What is amino acyl tRNA? A charged tRNA with a particular amino acid. So based on this codon, the charged tRNA is accepted here. So this step is known as alignment. So it is aligned, and now this, after this alignment, a transferring process is taking place. What is a transferring process? Here it has an amino acid. This tRNA also has an amino acid. So this tRNA present in the peptidyl region, that is the initial start codon. So this amino acid will be relieved from this tRNA and it will transferred to the amino acid present in the acceptor site. That means a chain formation is taking place, an elongation is taking place by the means of peptide bond formation. So by now the peptide bond formation is taking place. So one amino acid is here, this amino acid is relieved and it is attached to this. So a peptide bond formation is taking place. For this peptide bond formation, no energy is consumed because it is already prepaid while charging of the tRNA. So it is already prepaid. It is a strategic way of using the energy. So that energy is now utilized. So no energy is now consumed for peptide bond formation. It is prepaid. Okay. So for this peptide bond formation, in this 60S subunit of ribosome, the 28S rRNA present in the 60S subunit catalyzes this peptide bond formation. So you should remember that. So what is happening here? This amino acid from this is transferred here and peptide bond is taking place. So alignment and transferring. So this is the phase B of elongation. So now we have seen in the previous slide, likewise from the this transfer site, an amino acid is transferred to the acceptor site and the chain formation by the peptide bond formation is taking place and elongation is initiated. Now this elongation should be proceed further. It should proceed further. So for that, this ribosome should move through this mRNA and it should read all the codons present in this mRNA till the termination process is taking place. So for that, it should move like a zipper. So a zipper, a zip rider moves through the zip. Likewise, this ribosome should move through this, or it should go in this 5' to 3' direction on this zipper as like a rider. This ribosome should move. So for that movement, it needs energy. So how that energy is provided? So it will be done by some factors, elongation factors, along with GTP. It provides energy for this movement. So when this movement is taking place, this ribosome physically moves with the help of this energy over through this mRNA, like a zipper is moving as a zip rider is moving on a zip. So it will move and it reads the further codons. So the acceptor site will procure the next tRNA based on the next codon present here. It will procure the next tRNA with the corresponding amino acid and it will move here and it will give that amino acid to this chain and this chain will be elongated. And when this amino acid is donated here, this tRNA is deacetylated and it will become an empty tRNA, and at the exit site it will be ejected out of the ribosome and it will be recycled for the next protein synthesis. So this is the direction of ribosome movement. So you can also imagine this as like this mRNA is like a tape. So in previous days, that is in 90s or in 2000s, you would have heard about, there are some cassette players. So there are some audio cassette players, video cassette players. So basically they have a strip of information. You can see like a black strip that will be storing some audio or video information. It will be read by um, head, like a reader present in that video cassette recorder or audio cassette player. There will be a reader. That reader will read that particular that strip, video strip or audio strip, and it will decode the information from there, and it will send that information to the speaker as audio or to the TV as video. So likewise, the information present here, it will be decoded by the tRNA, that is the interface, and it will be it is producing the protein. So the protein is elongated. The amino acids are assembled here to form the desired protein based on this mRNA sequence, the codons present here. So if you see the energetics, that is the energy consumed for this protein synthesis, the major energy is, that is a high energy is spent for the tRNA charging, that is the activation. So it is a strategic way of using the energy. ATP is hydrolyzed here, and that here the prepaid energy will be later used for peptide bond formation. So there is zero energy cost at during the peptide bond formation because it is prepaid here. And the next energy utilization is in the initiation complex formation. So there 1 GTP will be utilized, and in the translocation step, that is way for the ribosome to be move further in the mRNA, another GTP will be used. So these are the energy spent while for protein synthesis. So protein synthesis is a high energy investment process. So it is a metabolically expensive process. So it will be done under extreme precision, strict quality control, and rapid mechanical movement require massive expenditure of cellular currency. So how the proofreading is taking place, as we have seen already, the tRNA with a particular amino acid, the DHO arm, it will recognize the particular amino acid residue. So what it does, it attaches the specific amino acid to that particular tRNA. So that acts as a proofreading step. So now this ribosome is moving here, protein chain is elongation is taking place, and now at a certain point it should be terminated. The factory need to stop the production and it should be shut down. So at a particular point. So how it is taking place? For that, like initiating codon, we have stop codons. So like UAA, UAG, UGA, these are stop codons. So this ribosome is moving here. When it encounters a stop codon, the stop codon at the acceptor site, it was encountered. So now it will release factor. So they attract the release factor. Sorry, they will attract the release factor, and this release factor will trigger the hydrolysis of this chain, and this chain will be released out, and this ribosome will be disassembled and it will be removed or moved away from the mRNA track, and it will be disassembled. The factory was shut down. So this is how the termination is taking place. So when stop codon is recognized, release factors are attracted, and that will hydrolyze the chain and disassemble the factory, that is a ribosome. So how this process is regulated? So the main regulation is at the level of initiation complex formation. So the EIF, that is eukaryotic initiation factor, that is the main regulatory factor. So if it is present more, it will do more initiation complex formation. So it will do this process. It will make the start codon to attach to the tRNA, and this initiation complex formation is easily formed because more amount of initiation complex is formed. When it is taking place in more amount, the translation process is fluently taking place in the it will proceed to the further steps. So this is the regulatory step for translation. So the presence of EIF, so this is the throttle or this is the gas pedal for this translation process. If it is present more, translation is taking place more. If it is less, translation or rate of protein synthesis is less. So certain drugs or therapeutic drugs are based on this translation as target. So tetracycline. So it will target the bacterial acceptor site present in the ribosome. So this will prevent the amino acid tRNA from binding. So the translation process will not take place in the bacteria, and the bacteria will not grow further. So it will be it is used as an antibiotic. Likewise, macrolides, it will bind with the 50th subunit of ribosome and it will freeze translocation. The ribosome cannot move further and the translocation process is not taking place and this translation is stopped. Puromycin, so what it does, it mimics the amino acid tRNA. So it enters the A site, the acceptor site, as if it is the original tRNA. It will enter and it block the acceptor site. So the chain formation will not take place. There will be premature chain termination. So that is the action of puromycin. Cycloheximide, so it inhibits the peptidyl transferase. So the peptidyl transferase is the enzyme needed for peptide bond formation while protein synthesis. So that will be inhibited. So protein synthesis will not take place efficiently. So there is a term called polysome. So we have seen a single ribosome is translating the mRNA and producing the protein. So what is polysome? We have multiple ribosomes reading a single mRNA at various sites. So this will improve the efficiency. So it will like a convoy procession. It will be arranged. It will be assembled in a line, and at various sites, various proteins are produced, and this will increase the efficiency of the protein synthesis industry. So that is the concept known as polysome. It is a macro efficiency. High scaled industrial mass production, likewise, in a single time, a mass production of protein will be produced. That is known as polysome. So like post-transcriptional modification, post-translation modification also takes place. The naive protein or the new nascent protein formed at the end of translation will undergo certain modification. It will undergo covalent modification of amino acid residues, gamma carboxylation, hydroxylation, methylation, glycosylation, cymogen activation, and very importantly, protein folding. Certain folding mechanisms will be done, and that will be assisted by some molecules called capirons. So that is those are the modification taking place at the post translation level. If this modification, some error is happening at the protein folding, that also will lead to certain disorders called protein folding disorders. So these are the post translational modifications. So in this video we have seen about translation. So first the step is charging of the tRNA, and it is binding to the small complex. So the small complex, it scans the mRNA. So initiation factor, that is the regulatory protein that helps in the formation of initiation complex. So first it binds to the small complex, and the next the larger complex also bound. So the entire initiation complex is formed, and we know that the larger complex has an A site, P site, acceptor site, peptidyl site, and a exit site. So the tRNA initially with the start codon that attaches to the start codon, it goes directly to the peptidyl site. Next, the acceptor site will add the next tRNA based on the codon present here. So that is the process called elongation. And after elongation is initiated, that is, it is aligned and the initial amino acid is transferred here, it should be moved, or it should be transferred or translocated through the mRNA series in the 5' to 3' direction. For that movement, GTP, another energy, will be consumed here, and elongation factor also helps for this. And next, by the peptidyl transferase, here the amino acids are transferred from the transfer site to the acceptor site tRNA, and this chain is elongated and the peptide bond is formed and the protein is formed. When it encounters a termination codon, this will be releasing factors will be attracted and the chain will be hydrolyzed and it will be released outside, and this factory will be disassembled and it will be leaving the mRNA chain. So how the drugs are acting? Streptomycin, it will distort the 30S subunit and this complex will not be formed. So translation is stopped. And how the tetracyclines are acting? It will block the 30S subunit and it will mimic the amino acid tRNA, and it will block the further tRNA binding to the A site. So it will block ultimately the elongation. So the this translation is stopped. What the chloramphenicol it does? It blocks the peptidyl transferase. Puromycin also it blocks the peptidyl transferase. So by these mechanisms, these drugs are used as an antibiotics. So after this translation, the nascent protein will undergo post-translation modifications. Thank you.